So how do synaptic vesicles actually fuse with the membrane? How do they get remade? And how is this entire mechanism sensitive to calcium? The SNARE hypothesis provides the molecular link between calcium sensitivity and vesicle release. The SNARE proteins are synapse-associated proteins that are highly conserved in structure and function across many vastly different organisms, suggestive of its evolutionary importance. The synaptic vessel is perhaps the best understood organelle, as all of its major SNARE proteins have been identified. Interactions between SNARE proteins on the vesicle and on the presynaptic membrane, modulated by calcium, lead to vesicle fusion. These membrane fusion mechanisms are essential for a variety of functions beside vesicle release in a neuron. Wherever there is a need for trafficking of molecules, whether it's for exocytosis between cells or trafficking nutrients between different organelles inside the cell, these SNARE proteins are present. Vesicle fusion in a synapse is catalyzed by the interaction of a vesicle-associated SNARE protein, or vSNARE, with other SNARE proteins on the membrane called target SNAREs, or T-SNAREs. Synaptobrevin is the main vSNARE, while SNAP25 and syntaxin are the main T-SNAREs. Other proteins bring the V and T SNAREs together, power their interactions, and separate them after fusion via a zippering mechanism, which helps them overcome an energy barrier. Calcium modulates the interactions between the SNARE proteins on the vesicle and the presynaptic membrane. Voltage-dependent calcium channels open when the axon depolarizes, allowing calcium to flow into the cell at the presynaptic terminal. A SNARE protein called synaptotagmin binds calcium and speeds the vesicle release. This calcium-dependent vesicle fusion mechanism is what allows neurotransmitters to flow into the synaptic cleft. Two toxins in particular, botulinum and tetanus, are enzymes that specifically target SNARE proteins, cleaving them and inactivating them. You might be familiar with botulinum. It's the main ingredient in Botox injections, used to paralyze muscles and reduce wrinkles. Both botulinum and tetanus work on the SNARE complex to inhibit the release of acetylcholine, an excitatory neurotransmitter that causes muscle contraction. Botox poisoning causes a total loss of motor function, in addition to youthful skin. Clostridium botulinum forms spores, which can be found in lots of places, including honey. Adult digestive tracts can eliminate them, but the digestive tract of newborns can't. The spores germinate, and Clostridium botulinum grows, leading to something called floppy baby syndrome, which includes hypotonia or reduced muscle tone. Tetanus toxin, or Clostridium tetani, has spores everywhere, which is why tetanus shots are required after stepping on a rusty nail. Tetanus is very similar in structure to botulinum toxin, but it selectively interferes with the release of neurotransmitters in inhibitory neurons. To summarize the key points of this lesson, neurotransmitters are released presynaptically in quantized amounts because they're released in discrete amounts by vesicles. Multiple vesicles fuse with the membrane, catalyzed by SNARE proteins, to generate a synaptic potential. Calcium entry couples depolarization of the axon to secretion via SNAREs. Finally, neurotransmitters activate channels in the postsynaptic membrane, which we'll investigate further in the next lesson.